Hi everyone, for those who don't know me yet, my name is Wei Yang. I'm your today presenter to explain our research proposal with the title Sex Identifications of the Canarian Odontophyllum, also known as the DABI, using the Random Amplified Polymorphic DNA Marker, the RAPD. So before we proceed to our research proposal, let me introduce shortly about our main point today, which is the Canarian Odontophyllum. The Canarian Odontophyllum is also known as the dabai, or you can simply call it as the Cebu olive. It is an indigenous and the popular fruit-bearing populations in the Borneo. This is because it consists of the high nutritional value and usually will serve as the food sources for the locals. For example, the locals will manufacture it to make the frozen puff and the fruit pickles. Besides, it is also an dioecious plants, which means it can only give rise to either male or the female trees. And in general, only the female trees are preferable to cultivated, while the male will be eliminated or being cutting down for other economical purpose. This is because the female tree can provide a more efficient fruit production. That's all from me. Now I would like to pass the presentation to our next presenter. Thank you.
Hi everyone, my name is Ernest and I will continue the presentation. Problem statement. Since it's not possible to differentiate Canarian Odontophyllum based on the physical appearance of embryonic form because it can only be identified after it reached its maturity, which is after 4 years. At first, seeds are sown and grow into seedlings. This means the general practices is highly recommended to plant Canarian Odontophyllum for 4 years and eliminate unwanted male trees to develop orchards that only have female trees to save space and labor costs. Thus, to prevent the uncertainty of the plant sex, the orchards retain only hermaphrodite trees. Next is hypothesis. The female-specific or male-specific diagnostic then will be obtained from the Canarian Odontophyllum through random amplified polymorphic DNA, which known as RAPD analysis, which the primer will be hybridized to the specific male or female DNA. After amplification of steps in polymerase chain reaction, both products from the amplification of male and female DNAs will be visualized on 1.2% of agarose gel. Hence, use the female-specific diagnostic then as a marker on the DNA ladder for the early stage sex determination. This is important than wasting time to wait the Canarian Odontophyllum for sex determination. Next is objective. We have two objectives, which are to identify the sex of Canarian Odontophyllum from young plant by applying RAPD PCR, and to advance the protocols of RAPD PCR. That's all for me, and I will pass it to the next presenter. Thank you.
Hi, my name is Jacyntha Ann and I would like to present about the literature review of Canarian Odontophyllum. It is defined as dabai or kambayau. The local names varies depending on the countries. For example, in Borneo it is called as Dabai, Dabe, Kambayau, and also Dabu, whereas in Sumatra it is called as Danau Majang and Katjai. For the distribution of Dabai, it is originated from Malaysia, Indonesia, Brunei, and Philippines. It is domesticated in Sarawak and was extensively grown in Cebu, Kapit, Limbang, and Sarike. The general morphology of Dabai tree is it has a maximum height up to 28 meters tall. It is in a medium tree size with straight trunk and huge stem. It is in round-shaped large pinnet with a crowded canopy, and the leaflet of Dabai is about 9 to 26 cm long with a width of 3 to 12 cm. The fruit of Dabai is in white color when immature and turn pink when matured, or even powdery black. The fruit is drupe berry with the flesh and seed and the size of 30 to 40 mm long and 15 to 25 mm wide. The uses and the economic importance of Dabai is it has high antioxidant capacity, the nutritious fruit rich in protein, carbohydrate, and the fat content. It has high content of minerals. Other than that, it also has high potential of economic exploitation because it can replace oil to make shampoo and soap. Next, the sex determination. It is a process of sex differentiation using different genetic concepts to decide a male or a female plant. Most non-dioecious plants, in a similar manner to that of mammals, have a sex determination system for XY with heterogamic and homogamic females. Next is the polymerase chain reaction. It is widely used in molecular biology. It will produce from a very small volume of DNA from 1,000 to million copies of DNA similar segment. There are 3 stages in PCR. The first one is denaturing stage, followed by annealing stage, and lastly, extending stage. Last, I will explain about the random amplified polymorphic DNA, or called as RAPID. It is a type of PCR reaction, but the segments of DNA that are amplified are random. It is able to differentiate between genetically distinct individuals, although not necessarily in a reproducible way. It is used to analyze the genetic diversity of an individual by using a random primer.
Good morning everyone, I'm Eylfenevaolen Jephen. I will be presenting the material and methodology part, and in this experiment there will be few steps will be carried out. The first step is plant material or the collecting sampling. So we will be collecting the samples from Sarike, Sarawak, 5 leaf samples from the male plant, 9 leaf samples from the female plant, and few samples from the young leaves. The second step is extraction of DNA. In this step, we will be using CTAB method, which described by Dolly and Dolly, 1987. All the samples will be ground into small pieces before centrifuge them. Transfer and centrifuge step will be repeated for several times until the DNA pellet is formed. The next step is preparation of agarose gel electrophoresis. In this experiment, we will be using 1.2% of agarose gel to run the DNA. So, we will be referring to a protocol introduced by Bin. The last step is PCR RAPD analysis. In this step, both male and female DNA samples will be run to see the differences between both DNA first, and the result obtained will be used as a marker in the latter to identify the sex type of the young leaves. So this is the summary for the whole material and methodology for our experiment.
Finally, the expected outcome of this research proposal is, first, successfully obtain the female-specific or the male-specific diagnostic band through the LAPD analysis. Then we can use this diagnostic band as a marker to successfully identify the sex type of the Canarian Odontophyllum. Then, finally, the optimization of the LAPD PCR protocol can be done. So it can be used instead of the sex identifications of the Canarian, but it also can use for other plant species. That's all from us. Thank you.